Hey guys, not really feeling my best today, so I'm just going to guide you through this lesson via the old school recording. Does this bring you back terrible flashbacks of remote learning? Sorry. So we're on cell structure and function, which is what you learn pretty much every year and then forget every year. So with biology, it's not just knowing and memorizing the parts and what they do, which you do have to do. It's being able to recognize them in a picture and identify what they do based on that and what type of cell they would be in. So we're going to go through these notes, fill in your guided notes as we go through it. All right, so organelles. We talked about these briefly on Friday. They're very small, meaning they're microscopic. We need a microscope to see them. They perform our various life functions Sternger, so synthesis, transport, excretion, respiration, nutrition, growth, reproduction, regulation. They're found in the cytoplasm. The cytoplasm's like that little jelly of the cell holding everything together. And they may or may not be membrane bound, meaning surrounded by a membrane of some sort. This is just a picture of an animal cell. Nope. It's complex. It's a eukaryote. Plant cell, remember, they tend to be more rectangular looking, also a eukaryote because of that nucleus and other membrane-bound organelles such as the mitochondria. Right, so the first organelle we're going to talk about is the cell membrane, which is also called the plasma membrane. So its structure and function are it surrounds the outside of the cell. So its function is it maintains homeostasis by controlling what enters and leaves the cell. Remember, homeostasis is how we regulate things, keep things in balance. It's also called the phospholipid bilayer because it's made up of two layers of phospholipids. This little circle with the tails is a phospholipid. There's one layer on top, one layer on bottom. We're going to get into that in more detail when we get to our cell transport unit. It's found in all cells, and then if we were going to draw it, we're all going to do really simple drawings here so you can recognize it. You would just draw a circle for the cell. This outer layer here is your cell membrane. Nothing to write for here, just listen. So if you're talking about a plant, it lies against the cell wall. So it's actually the inner layer here. The cell wall is the outer layer. And it pushes out against the cell wall to maintain shape, meaning it helps keep your plant nice and stiff. Next is our cell wall. The cell wall is there to support and protect a cell. It's found outside of the cell membrane, so that's this big rectangular structure you're seeing here. Note, not only plants have it, some bacteria have it as well. So it's found in plants, fungi, and bacteria. So note, us as animals, we do not have a cell wall. In a simple picture, you should have a rectangular shaped cell with an oval in the middle. That oval is the cell wall. Cytoplasm is jelly-like. It's where all of the chemical reactions of a cell happen, and it contains the organelles, meaning all of the cell parts, found in all cells. So whether you are prokaryote or eukaryote, if you are living, you have cytoplasm. In a picture, if you have a cell, you're just pointing to the inside stuff. That's your cytoplasm. The nucleus is large. It's circular. It's in the middle-ish of the cell. It is the control center. What that means, because control center is your middle school definition, is that it has DNA. DNA is the instructions for protein synthesis. So really important to remember, it's DNA there controlling where proteins being made. That's why it's the control center. It's in control of making all your proteins. This is found in eukaryotes only because it is a membrane-bound organelle. Simple picture, you have a circle, if this is an animal cell, in the middle you got a nucleus. The nucleolus, there's nothing to write, just listen. The nucleolus is inside of the nucleus. It makes ribosomes, and ribosomes end up making proteins. And again, it's in the nucleus. This part is in your notes, so mitochondrion or mitochondria, which is the plural, is going to be rod-shaped, so kind of like oval, and it has a double membrane, meaning it's surrounded by two membranes, and it has these little folds in it, and that increases surface area, so more reactions can happen. So if you didn't have these folds, reactions could really only happen on the edge, and it couldn't do its job very efficiently, but because we have all these folds, it can. And its function is respiration. Remember, respiration creates ATP energy. You breathe in oxygen, you eat glucose, and a whole bunch of enzymes are involved, and it ends up creating ATP, water, and carbon dioxide. Active cells like muscle cells have more mitochondria because they need the mitochondria to keep them moving. Sperm cells as well would have more mitochondria because they need to be able to swim to the egg. And this is found in eukaryotes, so plants, animals, fungi, and protists. All eukaryotes have a mitochondria. Sample picture, you've got your cell. You've got an oval with a bunch of little squigglies. It almost looks kind of like a little slipper. And again, nothing to write here, just listening. So mitochondria come from cytoplasm in the egg during fertilization. So that means all of the mitochondria you have, you actually got from your mom, not your dad. Chloroplasts. They're also rod-shaped, and they've got little stacks in them called thylakoids. Its main function is photosynthesis. Remember, synthesis means to make. So it uses the sun's energy, hence the photo part, to make glucose, which is food for plants. It's found in plants. Certain protists have it, but we'll focus in on plants for chloroplasts. Simple, sort of simple, I guess, picture. You've got an oval, and you've got these tiny little stacks. Think of them almost like little coins. Those are the thylakoids, and they're connected by these little lines. Those are called lamella. Ribosomes are just tiny little circles when you see a picture of a cell. Their job is protein synthesis, meaning their job is to physically make the proteins. So we consider these protein factories. And they're found in all cells. Remember, all cells' main goal is to make proteins. So both prokaryotes and eukaryotes need ribosomes. Simple picture, you have a cell, a bunch of little dots are your ribosomes. Vacuoles, they are fluid-filled sacs. There's a picture of one right here. They're used for storage, so think of them like lockers or your backpacks. They're found in animals, but in animals they're very small. And in plants have a large central one because they need to store that water for photosynthesis and to keep the plant healthy. So a picture, just putting it into perspective, you have a nucleus here. Your vacuole is just a big sac that takes up a lot of the cell in the plant cell. Nothing to write here. So in plants, they can also store cell sap, and they can also store sugars, proteins, minerals, lipids, waste, salt, water, enzymes. So really, they store pretty much anything that is necessary. A specific type of vacuole, and you can kind of see it working in this little animation in the background, is called a contractile vacuole. This is a special type of vacuole, and it regulates water intake by pumping out excess water to maintain homeostasis. So this is where things that live in fresh water need them because they're constantly getting water in them and they've got to pump it out so they don't burst. or lice, so it basically helps keep the cell from lysing or bursting. And it's found in unicellular protists, specifically Paramecium have them. The endoplasmic reticulum, which we'll abbreviate as the ER, is a series of flattened stacks that's next to the nucleus, so it surrounds the nucleus. The rough ER is called rough because it has ribosomes, so you see little dots on it. Smooth does not have ribosomes. This is where lipids are synthesized or made. Remember things like steroids. This is where transport of protein happens, so proteins get moved. And it can destroy toxic substances, kind of like a mini little liver in your cell. And these are found in all eukaryotes. Simple picture. You've got your nucleus, then you've got this little folded thing right outside of it. That's the ER. This would be the smooth ER because there's no dots on it. Golgi bodies are flattened membrane-enclosed sacs, and they're right next to the ER. They modify, sort, and package molecules that come from the ER for either storage or transport out of the cell. So think of these kind of like the post office of your cell, and they're found in all eukaryotic cells. So if you have a nucleus, you have a Golgi body. Simple picture, and I would draw it all. You have the nucleus. Outside of it, you have the ER, and then next to it, you've got these Golgi bodies. Lysosomes. These are small membrane-bound sacs, and they contain digestive enzymes. Remember, enzymes help break things down. That's what we're digesting here. And they can break down food, they can break down bacteria, and they can even break down worn parts of cells. So if you don't need a particular part anymore, then it will digest it. This is found in animal cells. Some plant cells do have them, but it's very rare. So we're going to stick to just animals for this part. Simple picture, just kind of a sac and then some dots in it. This isn't really one that you'd have to identify often on a diagram. Centrioles. These are paired structures that are near the nucleus. They're involved in cell division, so when the cell has many copies of itself, centrioles are used. And this is found in animal cells specifically. So only animals have centrioles. And then here's a little picture. They kind of look like little rigatoni tubes. Cilia and flagella. Flagella is a tail. Cilia are basically little hairs that surround a cell. They're used for movement. So if you're unicellular, flagella can help you move like a sperm. Cilia is just tiny little hairs extending that also help with movement. They're found in some plant and animal cells like sperm, and most single-celled organisms like bacteria, euglena, paramecium have either cilia or flagella to help them move around in their environment. Here's a sample picture. You've got a cell, these little hairs sticking out are cilia. The tail is the flagellum. Looks almost like a balloon. Just a picture of cilia and how movement happens with that in a paramecium. And over here, you've got sperm swimming. And again, that sperm has a lot of mitochondria in it as well because it takes energy to move that tail. This is just an example in your lungs. Our lungs have cilia, and they help move dust away from our respiratory system. An eye spot. Think of an eye. We use it to see. It's a sensory organ. This is a sensory organ that's used to detect light, specifically in euglena. This is a euglena here. So they have an eye spot at the anterior end, and it detects light. This is the anterior end right here, kind of near the tail, back. And this helps euglena find bright areas to gather sunlight and make their food, because euglena are protists that do have chloroplasts, so they can do photosynthesis. iSpot helps them get to that light to be able to do photosynthesis. Sample picture, here's your euglena, and then there's your iSpot right up. Then here's just a euglena spinning, or swimming, I can't speak. That's that euglena moving around. Alright. Now you're going to do your cell organelles and structures worksheet. There are certain things here that are labeled for you. So you're going to need colored pencils, a decent one. Obviously, we don't have lab stations here because we're in a trailer. But what you're going to do is you're going to write the organelle that matches the description, then pick a color to color it in both the animal and plant cell. Certain ones that you won't recognize are 9, which is the nucleolus, 14, which is the microtubules, and 16, which is the nuclear membrane. And if you have extra time, please go to Quizlet, study the cell structure and function set. And please remember to ask me for any help that you need, because again, I'm not going to formally lecture about this again. Thank you.